Hello and welcome everybody to my channel OmniBio Entrance. Today we will be discussing the next 2016 question paper. I will be discussing only the biology section. This section contains 15 questions. For questions 2.1 to 2.10, only one of the four options is correct. A correct answer will earn 3 marks and a wrong answer will earn -1 mark and an unattempted question will earn 0 marks. So the first question, a pedigree chart of a family with an autosomal recessive disease Z is given. Assume that inheritance is Mendelian and that all individuals with homozygous recessive genotype have the disease. Circles in the pedigree represent females and squares represent males. Filled spaces indicate affected individuals while unfilled spaces indicate unaffected individuals. If J does not have the disease or the recessive allele, the probability that I is a carrier of the disease Z is. Now this is the first generation, they are the parents, they don't have the disease. But they produce two offsprings, one with the disease, this is black one, and the other with I, that is without the disease. Now since there is a disease being expressed in the second generation, that is this black colored female, the parents must be heterozygous, that is they should have one capital Z and small z. Now what is the probability that I is a carrier of the disease? For the genotype, that is carrier of the disease means she should be heterozygous, that is capital Z, small z. For the genotype to be capital Z, small z, she should have received the Z allele, that is capital Z allele from either of the parent and a small z allele from the other parent. Hence there are three possibilities of receiving alleles from the parent. That is totally there are totally three possibilities of receiving alleles from the parent, and they have told that it is a carrier disease, so we have to calculate the heterozygous ratio. So two alleles I will have to have heterozygous conditions, that is capital Z and small z. So two alleles divided by the total number of alleles is 3. So 3 divided by 3 will give you 0.67. So answer A is correct. Which of the following pairs is an incorrect match? Transcriptional repression, gene regulation. Post-translational modification, phosphorylation. Cytoplasm, exon splicing of eukaryotic pre-mRNA. It is not exon splicing, it is intron splicing, so C is incorrect. Enhancers, increased transcription. Now a simplified model of the carbon cycle is depicted below. What are X, Y and Z? So Z consumes Y as well as X, and after decomposition it is getting converted into fossil fuels. Fossil fuels on combustion replenish the atmosphere with carbon dioxide. So Z must be some saprophytic organism, either fungus or bacteria. Then Y, carbon containing compounds in Y, they are so Y is being consumed by X, so X must be a herbivore and Y must be a producer. So because Y is replenishing carbon dioxide to the atmosphere through respiration as well as it is intaking carbon dioxide also, so Y is a grass, and X, it is releasing carbon dioxide to the atmosphere through respiration. So B is the correct option, that is X will be goats, Y will be grass, Z will be fungus. Next one, changes in certain parameters of the castor oil seed during germination in the dark are graphically represented. The curves 1, 2, 3 could indicate. So here the first curve is almost constant, so it must be the total dry mass curve. The second curve is increased, then it decreases gradually, so that must be lipid content. Lipid content, it will decrease drastically during the germination. So 2 must be lipid content and 3 must be sugar content. It increases up to a certain limit over here and then decreases. So answer will be C, option C, total dry mass, lipid content and sugar content. Next one. Students in a biology laboratory were asked to view certain cells microscopically and measure the size of a single cell. They were given a compound microscope with an eyepiece of 15x magnification and an objective of 10x magnification. The eyepiece has a scale with the least count of 10 micron meter. When the student focused the cells under the given microscope, they found that one cell measured 60 divisions on the eyepiece scale. What is the approximate size of that cell? So, they have given that the eyepiece count is 10 micron meter. That is, ocular division is 10 micron meter. And under the microscope, the cell is measures 60 divisions on an eyepiece scale. Hence, the size of the cell under the microscope will be 60 into 10. 60 into 10, that is 600 micron meter. Now they have given the magnification of the microscope, that is 15x into 10x, that is 150 times. Hence the original size of the cell will be 600, which you got by multiplying 10 and 60 divided by 150, that is 4 micron meter. Answer is option C, 4 micron meter. Then, 3 features of nitrogenous excretory products MNO of animals are tabulated below. Okay, the features of excretory products and MNO are given. The animals that secrete MNO as their major excretory product would be. So M, we will take it as ammonia because it is highly toxic. They have given toxicity as high. So we will take it as ammonia, and N will be urea. It is very low toxicity, and low will be for uric acid. So ammonia is highly toxic, urea is very low, and uric acid is slightly higher than urea. Then energy cost to produce. Then ammonia, less energy will be required. So low is correct. Then moderate will be required by uric acid, urea, and high will be required, high energy will be required for conversion of ammonia into uric acid. Then water loss to excretion. Ammonia is more water soluble than uric acid and urea. Hence water loss to excretion is high in ammonia, whereas moderate in the case of urea and low in the case of uric acid. So ammonia will be excreted by goldfish, urea will be excreted by rat, and lizards will excrete uric acid. So option D is correct. Next one. For the peptide sequence from N to C terminus, which of the following is the correct coding sequence of the gene encoding the peptide? So lysine is encoded by 2 codons, arginine by 5 codons, serine by 6 codons, cysteine by 2 codons, tyrosine by 2 codons, tryptophan by 1 codon, and histidine by 2 codons. So this is the sequence of the peptide that they have given in the question. Now we can deduct the mRNA sequence from this particular protein or peptide sequence. So lysine will be AAA, arginine will be CGC, and cysteine will be UGC, tyrosine will be UAU, again UAU, and tryptophan will be UGG, and histidine will be CAU. Now they have asked the question for the coding sequence of the gene. I have told in my previous slide that coding strand has got the same base sequence as that of the mRNA. Only thing is that uracil is replaced by thymine in the coding strand. So DNA strand will have the same sequence as that of this mRNA. That is, the coding sequence of the DNA strand will have the same sequence as that of the mRNA. So it will be having the same sequence, that is AAA, CGC, AGC, and in U it will be T. TGC, TAT, TAT, TGG, CAT, AAA. Now they have asked in the question from 3' to 5' direction. So option B will be correct. Next one. A glass plate is coated with a layer of agarose gel. This black colored thing is the agarose gel. A central well and 4 surrounding wells, MNOP, the MNOP, are cut on the agarose as shown in the figure on the right side. A mixture of X and Y antibodies is added to the central well. This is the central well, and there you have added a mixture of X and Y antibodies that react specifically with the antigens X and Y respectively. Well, M and N contain the antigen X. So M and N contain antigen X. Well, P and O contain antigens of unknown identity. All the antigen and antibody can diffuse through the agarose. The plate is then incubated for 48 hours. White lines of precipitate are formed between the antigen and antibody wells. These are the white lines of precipitate. Which of the following is likely to cause this? Now they have told that they have cut 5 wells on an agarose plate and they have placed antigens in MNOP wells and antibodies, that is a mixture, antibodies X and Y is added to the central well that reacts specifically with the X and Y antigen. So they have given M and N as X containing X as antigen. Now for P also, and after incubation for 48 hours, they have found some precipitation lines. Okay, for P and O, it is unknown antigen is present. We have to find out which one it is. Now since the lines X for N reacting with X antigen reacting with X antibody has given a line like this. X antigen from M reacting with X antibody has given a line like this. Similarly, P has also given a line similar to that of N and M by reacting with XY and antibody. So P also must be X because the line is to the reaction of M and N to XY. So P also must be X. Then O we have to find out. O is having a slightly different line from the other 3. So it must be Y. So answer is option D. Antigen Y is O and X is P. Then 3 promising candidate compounds X, Y and Z were analyzed in a drug screening project to identify a novel potent compound against lung cancer. The following graph show efficacies of these compounds in killing cancerous cell and normal cell. Based on the above data, which of the following has the best prospect to be a useful drug? So here, 3 promising compounds, that is X, Y and Z, were analyzed, and in drug screening they have used the compound X, Y and Z, and they have analyzed it with the cancer cell with the ability of the compound X, Y and Z to kill the cancer cell as well as to kill the normal cells. So before bringing a drug into the market to be sold as treatment for cancer, you will see it whether the compound or the drug affects the normal cells also. If it affects the normal cells, then it is naturally been not taken for as drug for cancer cells. It should specifically affect the cancer cells. If they affect the normal cell, then it is not a good drug for treatment of cancer. So here what they have done was they have treated cancer cells with compound X. So at 10 nanomolar concentration, X is inhibiting the division of the cancer cells. Then at 20 nanomolar concentration, compound Y is inhibiting or the division of the cancer cells, or it kills the cancer cells. At 30 nanomolar concentration, Z is killing the cancer cells. Then, but compound X has also got other defect, that is, it is killing the normal cells also, that is around at a concentration of, I will say 13 or 12. It is inhibiting the, or it's causing killing the death of the normal cells as well. Similarly, compound Y, that is 5, it is also killing the normal cells, but it is at a higher concentration, that is around 45 nanomolar concentration. It is killing the normal cells. Similarly, Z is killing the normal cells at a concentration of 40 nanomolar, that is the line 6. Although the compound X is found to be more potent against cancerous cell, it is not the best drug. Why? Because it is killing the normal cells at a slightly higher concentration, that is around 13 nanomolar or 12 nanomolar concentration. It is killing the normal cells also. Z is also not a good drug because at a concentration of 40 nanomolar, it is killing the cancer cells. But Y can be a good candidate for treating cancer cell. Why? Because at 20 molar concentration, it is killing the cancer cell, and only when the concentration goes up to 45 nanomolar, it is killing the normal cells, not at 20 nanomolar concentration. So compound Y will be the answer. So answer is B, compound Y. Then next one. Cladograms are branched diagrams representing evolutionary relationship. A cladogram with representative animals is shown below: salmon, salamander, lizard, mouse, chimpanzees. Now these M, N and O in the trait M, N and O would represent. Okay, M, N and O in a cladogram represent differences between the organism before and after that. So this before means for a particular character, before how it was, and when it got evolved, how the character changed. So let us take from the answer is option D. We will take from the answer itself only, then you will be able to understand what I meant. So M is lungs. M is lungs. Now salmon fish has got, do not have the lungs, whereas salamander, lizard and mouse and chimpanzees have lungs. So M. So M is here, that is we have taken M as lungs. Now lungs is absent in salmon fish. So it is before this particular point lungs were absent, but after this particular point M, lungs evolved in these species, that is salmon, salamander, lizard, mouse and chimpanzees. So M is lung. N is claws. Claws are absent in salmon fish and salamander, but claws evolved after the point N. That is in case of lizard, mouse and chimpanzees. So before the point N, claws were absent. That is salmon, fish and salamander, no claws were found. Then O. O is for fur. Fur. Fur is absent in salmon, fish, salamander and lizard. So before O, before the point O, the fur was absent in salmon, fish, salamander and lizard. But after the point O, mouse and chimpanzees developed fur. And P is opposable thumb. Opposable thumb is present only in chimpanzees, whereas salmon, fish, salamander, lizard and mouse don't have an opposable thumb. So answer is option D. M, lungs, N, claws, O, fur, P, opposable thumb. Now next question. For questions 211 to 215, one or more than one of the four options may be correct. Your answer is regarded correct only if you choose all the correct options and no incorrect options. A correct answer will earn 4 marks, a wrong or an unattempted question will earn 0 marks. In order to study lysine biosynthetic pathway in yeast, lysine auxotrophs. What are auxotrophs? Mutants that are unable to grow without lysine supplementation to the minimal growth medium. So, if the growth medium has got lysine supplementation, only then these auxotrophs will grow. Otherwise, they will not grow. So, lysine auxotrophs were screened. 4 mutants M1 and M2, M4 were isolated, which were all recessive compared to the wild type. To find out the number of genes affected by these mutations, haploid mutants were mated and the ability of the resultant diploid progeny to grow on medium lacking lysine were tested. The results are shown in the table below. Now here it is just given blank. Why? Because M2 on reaction with M1 is giving a positive. See here, M1 reaction with M2 is giving a plus symbol. That is, it indicates growth. So here also M2 with M1 will give a plus symbol only. Similarly, M3 with M2. See, we have chosen M3 here with M2 is a plus sign. So here also it will be a plus sign for M3 with M2. Here it will be a plus sign for M3 with M2. So what I have done was I have redrawn this graph and I have filled these spaces just like how I have told here. M1 M2 reaction is similar to M2 M1 reaction and it will be plus symbol. M3 M2 reaction will be similar to M2 M3 reaction over here. So it will be a plus sign over here. So I have filled all these places as I have told just now and I have put it in the next slide for easy understanding. So this is the first slide. We will come to option B. M1 and M3 are mutations of the same gene. M1 and M3 are mutation of the same gene. They show the same defect. See, M1 and M3 show the same defect. That is, here also minus, here also minus, plus, plus, M3 minus, minus, plus, plus. So M1 and M3 are mutations of the same gene. Then M2 and M4 are likely to display the same phenotypic defect. See, here also they both are same. That is plus here, minus, minus, plus, plus, minus, minus. So there are chances that M2 and M4 are mutations will show the same phenotype. Then M1 and M2 are mutation. M1 and M2 are mutation of the different gene. Of course, they are mutations of the different gene because they show completely different effects. This is not similar to this. This effect is not similar to this effect. So they showed completely different effect, and so M1 and M2 are mutation of different genes. Now coming to option A, there are 3 genes affected by mutation. It is not, that is also wrong. 4 genes are affected by mutation. Then next question. 2 unique reptilian species P and Q reproduce in ambient temperature range, that is 31 to 35 degrees Celsius only. A group of scientists found a strange observation regarding the male to female ratio as obtained from the fertilized eggs artificially hatched within laboratory incubators as shown in the figure below. The maximum lifespan of these reptiles is 5 years. 50 male and 50 female adults, each of species P and Q, were used to start a 10-year-long study. The eggs were always hatched in incubators during this study. In the absence of any other factors, which of the following possible outcomes is correct? First one, if the temperature of the incubator is strictly maintained at 33 degrees Celsius for 10 consecutive years, the species P is expected to have more males than in species Q. Okay, so if the temperature of the incubator is strictly maintained at 33 degrees Celsius for 10 consecutive years, the species P is expected to have more males than species Q. Yes, why? Because at this particular temperature, species P will have 50% males and 50% females will be there. So they will undergo reproduction and males will increase. But what is happening to species Q is at 33 degrees Celsius, there are no males nor females. So, no males but 100% females will be present, because they have given only the percentage of males here, not the percentage of females. But they have taken both male and female species for their studies. So here 0 males will be present means 50 per, the other half, the other 100% males, females will be present. So if the incubator temperature is maintained at 33 degrees Celsius, then species P is expected to have more males than the species Q. Then, if the temperature of the incubator is strictly maintained at 31 degrees Celsius for 10 consecutive years, there will be more males in species P than in species Q. At 31 degrees Celsius, you can see that there are 100% males and 100% males in the case of both the species, that is species P and species Q. There won't be any females. So, if the temperature of the incubator is strictly maintained at 31 degrees Celsius for 10 consecutive years, then P and Q will not survive. Why? Because there are only males in the population and no females, so reproduction won't be taking place. So option B is wrong. Then next one, if the temperature of the incubator is strictly maintained at 31 degrees Celsius for 10 consecutive years, then both P and Q will not survive in the lab. I told you already that P at temperature 31 degrees Celsius, 100% males will be present in P, 100% females will be present in Q. So there won't be any females and there won't be any reproduction taking place because females are absent. And so at 31 degrees Celsius, both the species will be wiped out. That is, P and Q will not survive in the lab. So option C is correct, B is wrong. Then next one, if the temperature of the incubator is strictly maintained at 31 degrees Celsius and 31 degrees Celsius for every alternate year for a total of 10 years, there will be more males in species P than in species Q. At 35 degrees Celsius, there are no males in case of species P, and there will be 100% females in species P. At the same temperature, there are 100% males in species Q but 0% females. Hence, if the incubator temperature is maintained like this, that is 31 degrees and 35 degrees for every alternate year, there will be more male species P than species Q, because in Q no reproduction will take place in the absence of females. So option D, if the temperature of the incubator is maintained at 31 degrees Celsius and 35 degrees Celsius for every alternate year for a total of 10 years, there will be more males in species P, because at 35 degrees Celsius males are absent but females are present. Here only 100% males are present, there are no females. So at this particular temperature, there will be more male species of P than species Q, because in Q no reproduction is taking place. So answer is option A, C and D are correct. Okay, before coming to the next question, I would like to explain what is stabilizing selection, directional selection and disruptive selection. In stabilizing selection, what happens is it preserves the average characteristic of the population by favoring the average individual. This is the mean of the individual that is favored. Whereas in directional selection, it will favor the individuals only in one direction. That is, it changes the characteristic of a population by favoring individuals that vary in one direction. So only the individuals at this extreme will be favored to grow in directional selection, whereas in disruptive selection, both the individuals with extreme characters in both the directions will be allowed to grow in disruptive selection. That is, these individuals vary on both sides of the mean. So they will be more selected for disruptive selection. Now coming to the next question. Over 50% of swallows living on a cliff in Nebraska were killed when a cold spell hit the area in 1996. Scientists collected nearly 2,000 dead swallows from the base of the cliff and captured about 1,000 live ones. By measuring the body mass of these birds, they found that birds with larger than average body mass survived. So the thousands which have survived had larger than average body mass and survived the cold spell better than the ones with smaller than average body mass. So the 2,000 dead swallows had smaller than average body mass. The data collected suggests the population is undergoing natural selection. Yes, because only one of the species is one of the species is only selected. That is the one with larger than average body mass is selected, and whereas the other one with smaller than average body mass is wiped out. The population is undergoing disruptive selection. Disruptive selection, if it was disruptive selection, then the species with larger than average body mass and smaller than average body mass would have survived. Then there is directional selection acting on the population. Yes, because it is selecting only one of the species, not the other one. That is, it is selecting only the one with birds with larger than average body mass than birds with smaller than average body mass. So it is selecting only one species, so it is directional selection. Then stabilizing selection is acting on the population. Stabilizing selection will favor the average, or will favor the one which is on the mean, that is a character somewhere between the larger and the smaller one, intermediate size will be favored, but they have not told anything about the intermediate size. So stabilizing selection is acting on the population is not correct. The diagram below shows the relationships among the midge larvae, larval salamander, and two species of crustacean Daphnia that inhabit certain pond communities. Study the relationships and deduce which of the following is or are correct. The dotted lines represent indirect interaction, and those which are not dotted straight lines represent direct interaction. Then presence of high number of larval salamander has an indirect effect on Daphnia rosea, and it is a positive effect. So larval salamander dotted line, so it is having an indirect effect, and if it is having a positive effect also. Why? Because when the number of or the population of the larval salamander increases, it will have a negative effect on Daphnia pulex, and so Daphnia pulex population will reduce, and this will favor Daphnia rosea population to grow. So larval salamander increase has got an indirect effect on Daphnia rosea. Now next one. When larval salamanders are present, predation reduces the population growth rate of the larger Daphnia, facilitating the coexistence of the two species of Daphnia. When larval salamanders are present, predation reduces. So when the larval salamanders are present, predation reduces the population growth rate of Daphnia pulex, and this will facilitate coexistence of Daphnia rosea, the and Daphnia pulex. So option B is also correct. Then the two species of Daphnia compete for the same resources, and hence increase in the resource availability will reduce the number of midge larvae and larval salamander. So when the two species compete for the same resources and increase in resource availability will increase the number of midge larvae and larval salamander, as both species of Daphnia have got a positive effect on the population of midge larvae and larval salamander. So option C is wrong. It will not reduce the number of midge larvae and larval salamander. It will increase the number of midge larvae and larval salamander. Then D. Increase in the number of Daphnia pulex will lead to a decrease in the number of midge larvae. Because increase in the number of Daphnia pulex will result in decrease in the number of Daphnia rosea because it is having a negative effect. So it will lead to a decrease in the number of midge larvae. So option D is also correct. So here the options A, B and D are correct. A, B and D are correct. Now next one. Assume that the coat color in mammal is detected by a genetically controlled biosynthetic pathway as depicted in the diagram below. The pigment gene X makes the enzyme X that catalyzes the conversion of the precursor to the gray pigment P1. So gene X produces an enzyme X that is required for the conversion of this precursor to P1. Similarly, the dominant gene Y produces the enzyme Y that controls the conversion of P1 to the black pigment. So P1 has to get converted to P2 means the gene Y producing the enzyme Y has to be present. Then on the other hand, the dominant allele Z produces an active enzyme Z that can completely inhibit the conversion of precursor to P1. Why? Because enzyme Z is having an inhibitory action on enzyme X. So the precursor, that is white color, will not be converted to P1 or the gray color. In all cases, recessive alleles respectively produce defective enzymes with no activity. So if recessive alleles are present in the genotype, they will have they will produce defective enzymes with no activity. All the three enzymes assort independently with no interference from other genes. State the color of the male with the genotype this and the female with the genotype this. So state the color of the male with genotype XX. It has got a capital X. So precursor white will get converted into P1, that is gray. But it is having the dominant allele Z. So what will Z do? Z will completely inhibit the action of enzyme X. So the male with this particular genotype will remain white itself because precursor is not getting converted to P1 due to the action of enzyme Z, which inhibits the action of enzyme X. And female with genotype small x, capital Y, small z. So female with this particular genotype, that is small x, it will not be able to convert the precursor to P1. So Y will convert P1 to P2. But since precursor is not converting into P1 itself, P1 is absent for the Y to act and produce the P2 color. So the female genotype also will be white only. So answer is option B. Both male and female are white. Thank you. Please like, share and subscribe. Thank you all for watching.